Human conception is a solitary process. A single sperm reaches the fallopian tube, penetrates the outer layer of an egg, and fuses its nucleus with the egg's DNA to create a 46-chromosome zygote. This single cell immediately begins rapid division, doubling into a growing cluster as it is propelled by tiny hairs toward the uterus. Roughly 5 to 7 days after fertilization, the cluster, now a blastocyst, implants itself into the nutrient-rich uterine lining. This diagram shows the standard architecture. The outer layer of the blastocyst forms a single placenta to draw nutrients from the mother, while the inner cells form a single amniotic sac to protect the developing embryo. Because the uterus is evolutionarily optimized for one occupant, a twin pregnancy forces the system to divide a strictly finite amount of space and resources. Clinical management of twins focuses on their architectural configuration in the womb rather than their genetic similarity. Fraternal twins occur when two separate eggs are fertilized by two separate sperm, meaning they enter the uterus as two independent seeds. Identical twins form when a single fertilized egg splits into two distinct cell groups during the first 2 weeks of development. The survival of these twins depends on their prenatal environment, specifically how much of the placental and amniotic structures they are forced to share. This is the DCDA configuration, the lowest risk setup. Each twin grows within its own amniotic sac, sustained by its own independent root base, or placenta. Fraternal twins always develop this way, starting as two separate embryos that implant in different locations. Identical twins only achieve this level of separation if the zygote splits within the first 3 days of fertilization. Splitting this early allows each half to develop its own outer and inner membranes, resulting in two entirely separate life support systems. While sharing physical space, these twins face no direct biological competition with one another. In this second configuration, the twins remain in separate amniotic sacs, but are anchored to a single shared placenta. This occurs in identical twin pregnancies when the embryo splits between days 4 and 8. By day 4, the outer layer is already committed to forming a single placenta. The embryos separate just in time to build their own inner sacs, but they remain tethered to the same nutrient source. This shared placenta creates a connected blood supply between the two babies. This setup allows for twin-to-twin transfusion syndrome, or TTTS. When blood vessels connect unevenly, one twin can inadvertently siphon blood and nutrients from the other, causing a dangerous growth imbalance. In this scenario, the timing of the split turns the womb into a high-stakes environment, where one twin's growth can compromise the other's survival. The highest risk arrangement occurs when twins share both a single placenta and a single amniotic sac. This configuration is the result of a split delayed until days 9 through 12, after both the placental and amniotic structures are fixed. Because there is no membrane between them, the embryos move within the same fluid space, which often leads to umbilical cord entanglement. As the twins grow and shift, these tangled cords can tighten, cutting off the oxygen and nutrient supply to one or both babies. Even rarer is the biological outcome of a split that begins after day 13. A split this late is incomplete, resulting in conjoined twins who share physical organs or tissues. The timing of this cellular separation determines whether the embryos develop into two independent individuals or remain physically connected. Medical management of twin pregnancies is dictated by these early cellular milestones. Obstetricians use early ultrasounds to identify the presence or absence of dividing membranes, which reveals the specific risk level of the pregnancy. Identifying a shared placenta requires frequent monitoring to detect the early signs of blood flow transfusion. When a single amniotic sac is identified, doctors often schedule early C-sections to prevent cord-related accidents in the final weeks. These critical factors, resource competition, the shared blood supply, and overall cord safety, are all determined by a biological arrangement fixed within the first 14 days of development.